This summer, I have had the opportunity to conduct extramural research at Cornell University under Dr. Mariana Wolfner to analyze the importance of zinc in the fecundity and progeny of Drosophila. Preliminary research has discovered that a release of cellular zinc, or zinc sparks, follows the trigger of egg activation and facilitates its completion. We wonder if similar processes occur in Drosophila and whether zinc is required for normal fertility. I like to start off my mornings in lab by sexing flies and separating them for our fertility assay, where we separate males and virgin females to be crossed with each other. This process involves knocking out flies with CO2 and separating them by sex. After mating, the male is removed and the number of eggs, larvae, and egg hatchability is analyzed in each of the 4 crossings. I compared the number of eggs and their hatchability between control females that were raised on a normal diet and experimental females that were raised on a diet lacking zinc. I also compared the egg number and hatchability for females mated to males that were raised on a normal diet or one without zinc. My results will determine whether zinc is needed for Drosophila fertility. Our next 2 processes involve targeting the FOI gene in the germline. FOI is the most highly expressed zinc transporter gene in Drosophila ovary. By targeting it, we can disrupt its function and analyze its effects on fertility. We will use 2 different methods to do so. In the UAS-Gal4 system, UAS is the target of Gal4 binding, and in this strain it is followed by a transgene expressing a small double-stranded RNA complementary to the mRNA of the FOI gene, which will target the mRNA for destruction. Once a fly with UAS RNAi is crossed with the fly expressing Gal4 in the germline, I will achieve RNAi knockdown of FOI in the germline of the offspring. My project tested 3 different Gal strains: MAT alpha, MTD, and NOS. The next experiment calls for a germline knockout, which utilizes CRISPR-Cas9 to knock out FOI in the germline. This starts with Gibson's assembly to create a plasmid expressing guide RNAs, or gRNAs, targeting FOI. We assembled the construct and verified the sequence and performed a maxi-prep to obtain plasmid DNA to be injected into flies to integrate it into its genome. Once we have those flies that can express the guide RNAs, we will cross them to another strain expressing Cas9 in the germline. In the germline of the offspring, we will achieve knockout of our target gene FOI. The effects of this mutation will be evaluated and documented to determine whether the FOI transporter is needed to regulate zinc levels in the egg during activation. If it is, then the knockout or knockdown females will be sterile. This experience has introduced me to amazing students and faculty, and I have come to have a greater appreciation for research. The environment at Cornell is very welcoming, and the mentors are very receptive to any questions regarding research, graduate school, and the environment in Ithaca. If you are thinking of conducting extramural research, choose development.